Hi, everyone. My name is Romy Amaro. I'm a professor of chemistry and biochemistry at the University of California, San Diego. And I'm here today to tell you about our work and all that we've done to use computational microscopy to study SARS-CoV-2. So the work in my group uses a technique known as molecular dynamic simulations. And I like to think of this approach as a computational microscope because that's exactly how we're using it. So what we can do is actually build highly detailed three-dimensional models of living systems or biological systems, and then explore the wiggling and jiggling of their many atoms using these numerical methods. So nowadays, what we can do is actually combine multiple different sources of experimental data. For example, these could be different types of structural data sets, such as cryo-electron microscopy, X-ray crystallography, and tomography, together with approaches and methods like glycomics, lipidomics, and genomics in order to build these highly detailed systems at the atomic level. And then all we're doing is essentially defining a potential function, which I'm showing here, which describes the interaction of all the atoms in the system. And then we're integrating Newton's equation of motion over time. And so we perform this numerical integration millions and billions and trillions of time steps. And we're able to do that on very large systems thanks to years of effort to develop wonderful community-based molecular dynamics codes such as NAMD, for example, which we've been running on the TAC Frontera and Oak Ridge National Labs Summit supercomputers. And so this is a massively parallelizable code that allows us to scale these calculations across hundreds of thousands of processors and get real performance gains. And so I'm really excited to tell you about how we use these data-centric simulation methods in order to pull together a whole bunch of different experimental data taken in different groups for SARS-CoV-2, and then extend our knowledge on what we know about the system. Okay, so by now, many of you are, of course, familiar with the virus known as SARS-CoV-2. So it basically looks like a golf ball with spikes sticking out of it. And those spikes are, in fact, called the spike proteins. And people care about the spike proteins for a few good reasons. So the first is that this protein sits on the outside of the virus, and it's the first point of contact that the virus has with the human cell. So it's a key component of the infection machinery. Also, it is a highly antigenic component. And so it is, as many of you probably know, one of the main components of the different vaccines that are now being gratefully injected into humans across the globe. So one of the key steps that has to happen in the infection process is that the spike protein, which I'm showing here in white, with little blue decorations on its surface, has to interact with another molecule or protein on the host cell or human cell. This molecule here is shown in yellow, and this is a zoom in of this interaction. The host cell receptor is called ACE2, angiotensin converting enzyme 2. And what happens is that the spike protein basically interacts with this receptor on the human cell via this little blue bit in the middle called the receptor binding domain. Or we also refer to it as the RBD. I'm going to keep coming back to the RBD in the talks today because our work has really shown a lot of interesting new features about this particular component of the SARS-CoV-2 virus and its interaction with human cells. So our work relies heavily on the beautiful structural data sets that have been deposited into these open data sources. In particular, the first study that we really relied heavily on was one that was deposited actually in February of last year of 2020 by Jason McClellan and collaborators at the National Institutes of Health. And this data gave us a detailed picture of what the spike protein's head actually looks like. And you can kind of see that here. One of the interesting features of this system was that this little green bit at the top, that was how they colored at the time, the RBD, or that receptor binding domain. They saw that it was in the or open conformation. A few weeks later, another study by the group of David Wiesler at the University of Washington presented another study that largely recapitulated the same structure of the upstate, but they also developed a model of what the closed or downstate would look like. And so we took both of those structures and computationally filled in different loops and missing gaps, if you will, and we're able to create a model of the whole system. And you can see those here. So these are the complete models that were only possible to make with these computational methods. So the other thing that is really important for these systems that experimentalists can't see are something called glycans. And so glycans are what we call a post-translational modification. So basically we have the protein, the spike protein, and then it's decorated with essentially a coating of sugar. And it does this in order to hide itself or mask itself from the human immune system. So it turns out that our cells, our human cells, are also coated in these sort of sugary molecules. And so the virus, by decorating its foreign proteins with this sugary shield, actually, it's a mechanism that it uses to disguise itself from the components in the immune system that would otherwise recognize it as a foreign agent. So we actually were able, we used these glycomics data sets that were available from the group of Max Crispin, together again with Jason, and the data from Paris Suizadi at the University of Georgia, to build out at each of these different places on the spike protein what those sugars look like. And what was really cool about this is that experimentally they cannot see what these sugars look like. So this was a very a place where HPC has a lot to give, because it allows researchers to actually get views into this system that otherwise would be inaccessible to experimental techniques. So this is what our full spike protein actually looked like. So we have all of the domains. A large component of that comes from the cryo-EM, some of which we had to model. We had two different versions of the system, one that had the RBD in that up conformation, another within the down conformation. And we have all of our glycan molecules. You can see how they basically decorate the entire length of the protein. We also have the viral membrane with a realistic lipid, phospholipid composition. And this is what our system looks like at the end of the day. And then what we're doing is essentially integrating Newton's equation of motion. We're basically able to animate the system, or to look at the wiggling and jiggling of its atoms. And so the wiggling and jiggling that you see here, these are all of the different atomic configurations that are predicted across a microsecond of dynamics for this system. And here you can see some technical details. These systems have about 1.7 million atoms. They're also covered in water and ions, but I'm not showing those because otherwise you can't see the protein. We use the Charm 36 force field, and we're able to run NAMD2 on the amazing TAC Frontera supercomputer, where we had performance benchmarks of about 60 nanoseconds per day on 256 nodes. So it was really fantastic to have quite generous access to this system. That was really key for the discoveries that we made, which I'm about to tell you about. And I'll just mention we were able to get about 4 microseconds for each of the systems that we studied. So what did we learn? So one of the reasons why people were really excited about our work is because we showed the world for the first time what the fully glycosylated spike protein actually looked like. So as I mentioned, the experimentalists can't see the sugars. So on the left is actually a picture of the spike protein, as if this is what the structural biologists see. So this is what the experimental structure techniques tell us. They show us a picture that looks like this light blue image. This is the membrane on the bottom. On the right is actually what the spike protein actually looks like if you consider that sugary coat or that glycan shield. And you can see right away, it's very evident. So here the sugar molecules are shown, the glycan shield in this dark navy blue. It's just so obvious that it looks very different when you actually consider those glycans there. And so each of these little sort of puff balls or little tufts of fur represents the movement of one glycan and sort of how it's rapidly dynamically moving around in space. And when you have all of these sugars on the outside of the protein doing this, they create this very effective mask or shield that hides the protein from the human immune system. And a lot of people are interested in this, not only to know what the shield looks like, but also because holes in the shield You can think of them as sort of vulnerabilities of the shield, or chinks in the armor, if you will, of the virus itself. And so we can think about developing small molecule drugs that will actually go into these open spaces and sort of break the system and stop it from working. So our simulations actually showed why the spike actually opens and closes, or why it has that up and the down conformation. And this is so cool. So it turns out if you look at the closed spike, which has that receptor binding domain in the down conformation, what you see is that the spike has basically hidden the receptor binding domain. Again, I'm showing that here in this light blue. It's fairly well hidden underneath that shield. And here's a view from the side. You almost can't see it at all, just a tiny little bit sticking out. That's in contrast. If you look at the open spike, where that receptor binding domain is in that up conformation, you see it presents a completely different face to the ACE2 receptor. And this is really important. This light blue bit, as I mentioned, is the key part of the spike protein that has to make the handshake with the human cell. So, and here from the side, you can again see how dramatically different this system looks when you're thinking about or considering the glycans in these two different sort of conformational states. And so, it's funny, when the experimentalists first characterized the system, when they saw the structures without glycans, basically what they see is up and down, or open and closed. But, I often think that if they had been able to see the glycans in their pictures, or that they were able to capture using cryogenic microscopy, that they instead would have called this the shielded mode and the exposed mode, or the defending mode of the spike, and the attack mode, because it has to be in this up and exposed conformation in order to actually infect the cell. So the other thing that was really interesting that we noticed when we were looking at the system is that when we rebuilt those sugar molecules, there were two in particular at positions here, which I'm showing in blue and green, which actually appeared to fill in the void space under that opened up underneath the receptor binding domain when it went into the up conformation. And so it occurred to us that these sugar molecules may actually be acting as sort of a structural scaffold. In fact, they look sort of like a kickstand on a bicycle, that they would help prop this domain up in the open conformation and thus help infection. And so we also created in silico mutations where we converted these residues to alanines. And when we did that, it removed the glycans at these sites. And we ran more dynamics of this mutated system. And when we did that, what we saw was this domain actually collapse down. And our prediction would be that if that receptor binding domain collapsed down, it would be hidden by the glycan shield, as I just showed you, and the interactions with ACE2 would be decreased. And so at that point, we decided to put these predictions to the experimental test, which is really the ultimate the ultimate test for all of these computational methods is, is it really happening in the real world? And so we called up Jason McClellan, who's turned into a fantastic collaborator at the University of Texas, Austin. He was, the lead author on the very first structural papers of the spike, and has a long history, an important history of studying these coronavirus envelope spike proteins. And they created a new experiment that would allow us to test these mutations. And basically what this data tells us is that, in fact, the interaction of the spike protein in the two mutant forms, where we've actually gotten rid of those glycans, actually it supports our hypothesis that the structure collapses into the down position and the interaction with ACE2 is decreased. And so this work was published in October of last year in a wonderful journal called ACS Central Science. And, again, we're really excited about it. Just to recap, because simulations actually established, so beyond just showing the shielding, which we were all interested in knowing, many scientists around the world, these simulations also established a never before seen role of glycans in biomolecular systems. And that was that this study actually showed for the first time how these two glycans in particular actually acted not only to shield the spike, but as part of the infection weaponry itself by locking and loading the spike for infection. So another So that was just the first study, and we've continued to, as you can imagine, we're very interested to continue these studies. One thing, one of our next steps was actually trying to understand the full functional transition from the closed to the open state. So instead of just simulating the two end states, could we actually get an idea using molecular simulation of that opening pathway? But the problem is that it's very slow. It's a seconds. It's a times. It's on the seconds time scale in terms of how long it actually takes. And as I just showed you, we were looking at microseconds of dynamics. So there's many orders of magnitude that we're missing or we're typically unable to access using conventional molecular dynamics. So we partnered with Lillian Chong at the University of Pittsburgh, who turned into another fantastic collaborator. And she has been developing for a number of years a technique called weighted ensemble molecular dynamics, which focuses specifically on these functional transitions. And it does that by essentially reducing the amount of time that we spend sampling conformations that we really don't care so much about, and instead encouraging the system to go over or go along a particular what we call progress coordinate. In our case, that progress coordinate was sort of just, it was a distance of how open the system was becoming. And so we were able to, again, with generous access to the TAC Longhorn machine, we were able to run on 100 NVIDIA V100 GPUs for a few weeks and generate, actually, this is wrong, it's more than 100 terabytes of data. We generated about 250 terabytes of data. And in the process, we set a new high watermark for the size and complexity of systems that have been studied with weighted ensemble methods. I'm showing you here, that's why we gave it a little crown. And just to give you a sense, so if we wanted to use the conventional molecular dynamics that I told you about in the first story, in order to sample that opening transition, to get even one opening transition, we estimate it would take about 800 years to do that using ordinary or conventional molecular dynamics. This is in contrast to the weighted ensemble MD. With her technique, we were able to generate over 300 opening pathways in a matter of a few of 2 to 3 weeks, which was really awesome. And of course, it's this boost in efficiency, in fact, many orders of magnitude more efficient than standard MD, that really is an important advance for high performance computing in the space of molecular simulation with high impact to public health. And so this is a movie of what we actually saw. So we, again, we're simulating that closed to open transition where the receptor binding domain goes from the down to the up state. And I'm showing here again sort of the same imagery of the protein with various glycans in dark blue and the receptor binding domain in light blue. And there was one glycan in particular that I'm showing in hot pink that jumped out. And one thing that was really interesting about this was if we were just, this was a glycan that in the first set of studies, when we weren't looking at the opening pathway, but where we were just studying these two endpoints, it doesn't look like this residue is doing anything interesting. But when we actually look at the opening pathway, we see a completely different story. And that is that this pink glycan at position N343 plays a very important role where at the beginning it's helping to shield. But as the receptor binding domain opens, this residue does sort of like different hand jive type motions with other residues on the receptor binding domain and actually helps to kick up and out that receptor binding domain to achieve the up state. So this was a really interesting prediction, which again, we went back to Jason McClellan and they ran another set of experiments, not only mutating this particular glycan, but a series of other residues that we predicted would play important roles in this opening pathway. And they showed, in fact, a quite dramatic reduction in the ability of the spike to open and interact with ACE2 when we remove this glycan. So this was really cool because we showed essentially that this glycan acts as a gate that controls the opening of this really important pathogenic protein. So many of you have heard, of course, about the different mutations. And as you can imagine, these methods are also very handy or skilled at allowing us to give rapid assessments of the effects of these mutations and variants of concern. And I just want to highlight one study that we carried out together again with Jason McClellan and Rino Rapulli, who is a fantastic investigator in Italy. And here they have discovered a variant of the virus that has an really sort of extreme mutation where it's actually inserted an 11 amino acid stretch of residues, as well as introduce a new glycosylation site. And what I'm showing here are our predicted models, where not only do we have the insertion modeled in yellow, which completely remodels this important region that otherwise would be interacting with neutralizing antibodies. But here I'm showing where in gray, I'm showing how a neutralizing antibody binds to this site ordinarily. With this insertion and a glycan modeled, and again, you can see this glycan here in dark blue, you can see how inserting a glycan in that position basically completely obliterates the ability of these neutralizing antibodies to bind to that site. And so this is an escape variant, as we call it, because it actually, this virus was not susceptible to being neutralized by potent polyclonal patient sera. And so beyond just looking at the spike, we're also very interested, of course, to look at the other components more closely in terms of what it's interacting with as it approaches the host cell. And so we have carried out simulations of that ACE2 receptor. One of the interesting things we found here is that it actually undergoes, it has a very large and sort of unexpected degree of flexibility relative to the membrane, and actually can lean down and sort of touch the membrane. And we think it's sort of this mechanical flexibility actually really helps to facilitate the membrane fusion that must occur in order for the virus to actually infect the human cell. And so, so putting it all together, we're working towards really trying to understand in all atom detail how this molecule works, so how this very important spike protein actually infects the human cell. And, in doing so, we can create new ideas for potential therapies. And one of the really cool things that we've done also is develop a model of the interaction of the full-length spike protein with ACE2 across a 2 parallel membrane system, and actually used an AI-based method called Deep Drive MD, which is developed by Shantanu Jha at Rutgers and Arvind Ramanathan at Argonne, in order to help increase or accelerate the conformational sampling that we're seeing. And in fact, we saw a remarkable degree of stalk bending, which is a finding that has been supported by others, and where, again, it could be possible that we could develop therapies that might block that flexibility and thus decrease infection of the human cell. And then the other thing that we were really proud of that we were able to achieve as part of our Gordon Bell submission was actually the development of the full-scale viral envelope. And here what we've done is actually taken tomography data and used a multi-scale approach to fit in high-resolution spikes into that picture. So what I'm showing here is the full all-atom virus model of SARS-CoV-2. It has over 300 million atoms and sets a new high watermark for computational virology in terms of the size and complexity of systems studied. And here we again, really relied heavily on the wonderful architectures such as Frontera to do a lot of our prototyping work. And then we also ran the full-scale dynamics on Oak Ridge National Labs Summit machine, and had really fantastic scaling there to systems that took nearly, it can take nearly the whole machine. And so this was the Gordon Bell team. We were very fortunate actually to be selected as the winner for the Gordon Bell Special Prize in COVID-19 research. And I just want to, express my gratitude to this fantastic team. It was almost 30 people, 11 institutions, 4 of the best supercomputing sites in the country, together also with NVIDIA, and a fantastic collaborator in the UK. And anyway, it was really, I think, a wonderful representation of what teamwork can do and achieve, when we all work together towards a common goal. And on that note, I also want to say how SARS-CoV-2 or COVID-19 has presented the research and science world with a completely different new mode of operation, where worldwide collaboration and sharing has really jumped to the fore as being such an important part of the ethos of all of the work of ours and many others. And I was really sort of heartened by our field's response to sharing of data and methods and models together in ways that we've never done before. And that has been really fantastic. And it also led to the development by the NSF Molecular Sciences Software Institute, MOLSI, this wonderful new resource. It's the COVID-19 Molecular Structure and Therapeutics Hub, where researchers from all around the world have now been depositing their models, their methods, and their data sets so that we can share openly and learn from others. And I think particularly with artificial intelligence-based methods, over the longer term, this is going to be a very important resource for new studies that where there's really just unimaginable impact yet to be found there. I also just want to emphasize how important it's been to have high quality community software that's open access, such that any researcher around the world could help in the fight against SARS-CoV-2. And particular want to give a shout out to VMD and NAMD, who've just are fantastic scientific teams who have provided the community with such really terrific software for so long, and really grateful that they exist. And I want to just emphasize how important it is that we continue to support them. And then lastly, also, the other thing that was really unique about this whole situation was the COVID-19 High Performance Compute Consortium, which came online in March of 2020 and has provided our field with an enormous resource. And this was really a global effort that is awesome. And I know that I believe that they're sort of hardening this sort of approach towards readiness and resourcefulness in new ways, so that they can pivot the next time we have some type of global crisis. We will be even more ready. But this has been a really fantastic use of resources, and very important for the success of this work. And with that, I'd like to thank you for your attention. I have, as I mentioned, I've already shown one acknowledgement slide. There were so many people who made this work possible. In particular, Lorenzo Castellino, Ziad Gayeb, Abigail Dahmer, Tara Stain, Shirley Ahn. Adrian Mulholland's been a key collaborator throughout all of this. Elisa Feda, also Jason McClellan, Carlos Simmerling, so many people. So I'd like to thank all of those folks. I couldn't say everybody's name, and also the funding agencies and the compute sites for all their support.